Namaste students. हमेशा खुश रहो बिटिया. I am JV's Dr. Neeraj Dholia, Faculty of Agriculture and Veterinary Science, Jayoti Vidhyapeeth, Women's University, Jaipur. So today we are here to discuss your practical session. So the practical is study of TS of testis, and this comes under the subject developmental biology. Okay, so I'll share a screen here. So what is the anatomy of testis and what is it? So testis is a primary reproductive organ of male. Okay, so primary reproductive organ means it will produce the gametes. So this will produce the male gametes in the male. So it is the primary reproductive organ. So for performing this practical, you need a compound microscope, and if it is a digital microscope, then very good. You can take the images and for the result purpose. Along with the compound microscope, you need permanent slides of the testes of human or mice. Okay, so you need tissue paper. So for cleaning the lenses, for cleaning the slide. So what is the procedure for performing this practical? First of all, you will clean the lenses of the microscope, you will clean the slide, and then you will fix the slide on the microscopic stage, and you will focus it, and you will observe what you have you are seeing in the under the microscope, and you should correlate the structures you are observing with the theory you have studied in your subject theory session. So the testes of mice or the human, it comprise several seminiferous tubules that is embedded in the interstitial tissue. So a thick fibrous tissue called tunica albuginea cover the testis. Okay, and then it comprises different types of cells from the outside to the lunar in the manner that is the spermatogonia, the spermatocytes, spermatid, and spermatozoa. There are some Sertoli cells are also there between the germinal cells, and also the leading cells. Leading cells are there that will produce the testosterone and present in the interstitial tissues. So in the process of spermatogenesis, there are three processes, three phases, like multiplication phase, the growth phase, and the maturation phase. Okay, so the phases you have already studied in the theory session, so we will not discuss the phases here, and we will focus on the anatomy of the tissue. So this is the anatomy of the tissue, the testis. This is the epididymis part, body of epididymis, the tail of the epididymis, and the head of the epididymis. These are the seminiferous tubules where the production of the male gametes occurs. This is the layer of the tunica albuginea. Okay, so you will observe the structure under the microscope, and the precautions you should take during performing the experiment, that The microscope should be handled with care because it is very delicate thing, and the lenses are very sensitive, and adjust the lenses such that the focus is better. So more the focus is good, the more you get the clear images. So you observe and take the images under the digital microscope and put in your observation and results portion. So thank you, and hope you are satisfied with this detailed session. And if you have any query, please write in the comment box. And this session is powered by digital version 2 Jyoti Vidhyapeeth Women's University, Jaipur. Thank you.